So the story that I would like to tell you about, a search for the origin of our adaptive immune system, began with a collaboration with a very noted immunogeneticist, Jan Klein. Jan and I shared an interest in phylogeny of the immune system. He was particularly interested because he wanted to know the origin of transplantation genes, MHC, major histocompatibility genes. I was interested in particularly trying to figure out which came first of these two lineages that Jean-Claude mentioned that are the key to our adaptive immunity, which the T cells or the B cells, which actually arose first in evolution. And so we joined forces, our lab, together. We knew at that time that all jawed vertebrates, from the work of many different investigators, including my mentor, Bob Good, and Louis de Pasquet and others, that we all share basically the same kind of immune system. We have a thymus where we generate T cells and where they rearrange their receptor genes to generate diversity of receptors. They have B cells that are generated in hematopoietic tissues. They have major histocompatibility genes, class 1 and class 2, where intracellular and molecules are cut into small pieces and presented by these cell surface MHC 1 and 2 proteins. And they have the genes that are there for rearrangement, essential for putting together the different gene segments that cause the development of diverse T cell receptors and B cell receptors. We couldn't go back to some of the earlier jawed vertebrates because they no longer exist. We know them only through fossil remains. And as Jean-Claude mentioned, they're the jawless vertebrates. Lampreys and hagfish are the only extant vertebrates of the jawless lineage. And so our studies began with the lampreys, and in particular a species. There are about 40-something lamprey species, and they are worldwide in their distribution. About half of them are predators, like the sea lampreys that we have studied most. They spend much of their life, up to around 4 or 5 years, as filter feeder larvae, then undergo metamorphosis, becoming larger and developing a sharp ring of teeth, and predators of fish. They then breed and pass away, and the cycle starts over again. It's a difficult, complex life history, and it's not been recapitulated in culture, so all of the study animals are caught in the wild. We began by looking at cells in a hematopoietic tissue of the lamprey larvae, because Jan and his colleagues had identified a gene, a transcription factor that's important in hematopoiesis and in B cell development in particular. They found in looking to see which cells express the transcripts for this gene that small round cells in a hematopoietic tissue near the intestine was a site where these cells develop. I thought we might be able to isolate these cells on the basis of their physical and chemical composition in a simple way. So we used flow cytometry to separate or isolate a population of cells that looked like our lymphocytes, even by high-power electron microscopic thin sections. But this cell could have been any kind of cell. Our strategy was to create, to look at the genes that they were expressing, or to sample them by making a cDNA library and then getting, at that time, what was high-throughput sequencing, we could get the sequences of the transcripts or the cDNA for about 1,000 genes that we annotated our catch by going through the databases that were available at the time, and this was in around 2002. And we found that they expressed lots of the genes that our lymphocytes express for migration, for activation, and so on. What we didn't find were these classical genes that for the adaptive immune system, the MHC class 1 and 2 genes I mentioned, T-cell receptors, antibody or immunoglobulin receptors, or RAG1 and RAG2, the genes that initiate the rearrangements that start the assembly of T and B cell receptors. So we went back and got more sequences, and we still didn't find these. So, but it had been reported that in the 60s that lampreys could respond if you immunize them with foreign red blood cells from humans or sheep. They made something in their serum that agglutinated or clumped specifically the red cells, the foreign red blood cells. But no one had been able to define what these agglutinins actually were. So we thought there must be something for specificity. So Zev Panzer and I, who had joined my lab then, decided to, if we could stimulate these lampreys, and we might, in effect, catch them in the act of responding. And so we immunize them with live E. coli bacteria, sheep red blood cells, PHA and pokeweed mitogen, plant mitogens that stimulate proliferation of T and B cells of mammals. And when we did that, we generated a group of cells that had the light scatter characteristics of these larger cells. So they indeed looked like lymphoblasts. We made a selective library, cDNA library, from these lymphoblast cells, and we got several sequences. And they all turned out to be coding leucine-rich repeat proteins, a very ancient type of building blocks that is used by every living thing on our planet for various reasons. It's a very adaptable unit. They expressed a summer student wishing to learn how to do some molecular biology, sequence these leucine-rich repeat sequences that we had, and we were shocked to see that every one of them was different. They had different numbers of these green blocks that are 24 amino acids each, and they all had this N-terminal and C-terminal LRR module, an LRR1 and a short connecting peptide. But the sequences, particularly beginning here in the N-terminal and going to the C-terminal, turned out to be very different one from the other. They also had an invariant region, or a constant region, a stalk region, which predicted that it would be tethered to the surface. We expressed this in lymphocytes from a mouse thymoma cell line, and it indeed came to the surface. We named these variable lymphocyte receptors because they were expressed by lymphocytes-like cells and also were so highly variable. Just to give you a flavor of the variability of these two at the top are closest in sequence to each other, and yet they have about 10% difference in their sequence, and so on around this cartwheel phylogram. One other feature of this is that there were rarely duplicates, and when we did the assay on a single cell analysis of the transcript, they only expressed one VLR allele and not the other, implying that each lymphocyte would only have one type of receptor. We were curious as to whether these multiple genes were encoded by these multiple variable lymphocyte receptors had lots of different genes encoding them, or some other basis for their diversity. And so we found that Chris Amamiya had a genomic library he had made from sperm of a sea lamprey that we were studying. And so we used his library with probes of our VLR genes to go back, and we didn't find multiple genes encoding them. We only found one VLR gene, and it was very incomplete in that it encoded for parts of the N-terminal and C-terminal and the long stalk region, of course, that's invariant, but not the rest of it. These were separated by long stretches of intervening non-coding sequences. When we were able to go in and look at his cDNA library using our probes, we eventually found that there were flanking this incomplete VLR gene, there were coding sequences for leucine-rich repeat that if somehow stitched in to fill in this intervening sequences and the non-coding sequence deleted, could account for the kinds of mature variable lymphocyte receptor gene that we were seeing the products are. And we found that these lymphocyte-like cells were the only ones that did that rearranged or assembled these mature VLR genes. Other types of cells, like red blood cells or erythrocytes, had this germline configuration for this VLR gene. We've learned more about how that process of assembly actually works, but we still don't know all of the fine biochemical steps or molecular steps of the process. But we know that they're flanking this incomplete germline gene are over 400 different cassettes or sequences, coding sequences for the pieces that are needed to complete a mature VLR gene. And these can be brought in on the basis of a short stretch of nucleotides, maybe as few as 18, to match the recipient acceptor, and that holds them together, apparently, for copying in of a piece of these donor sequences. They have stop codons in them and other problems, so they're not used in their completeness. So that process is repeated over and over and over, this stepwise assembly process, until you get a mature VLR gene. The process can begin on either end, 5' or 3' ends, and go through to completion. And it only happens on one allele and not the other. So there's still things that we don't know about it. An important difference of our VDJ recombination to make our T cell genes and antibody genes is that these different components, these cassettes that are flanking, aren't moved. They are merely serving as templates, and they're copied in. So it's not a rearrangement of the gene in and of itself. Now, we couldn't look at the antibodies easily. We had no clones of malignant clones that were making the antibodies, but Brant Heron in our group devised a way to make monoclonal hybridomas or monoclonal antibodies of this VLRB type. And using those monoclonals, we could look by negative negative image electron microscopy to see that they form this flower-like configuration. This cartoon explains what we think these images actually look like. The stalk regions themselves are flexible, so it's not always a perfect tulip shape. It can be almost any shape. I didn't point them out, but there are several cysteines at the carboxy terminus, and these are used to form disulfide bonds to link together these chains, each of which has an identical sequence, and the variable lymphocyte receptor genes, the diverse part, is here. Now, the first one, monoclonal that Brandt made, Byung-Won Hong in Ian Wilson's laboratory, was able to form crystals and determine the structure of this antibody called RBC36, this variable portion of it, how it binds to its antigen, which is three sugars put together in a polysaccharide or H-trisaccharide. This is the epitope or the antigen that is used to type our blood group O positive. And you can see that it is caught in here in this little cleft between this loop, which I didn't mention before. That's encoded by a highly variable insert in this carboxy terminal LRR portion of the gene. These dots show hydrogen bonds, water dependent, with this LRRV2, one here, two in this N LRRV, and then several important junctions or connections with this loop, this variable loop. So it looks sort of like this is a solenoid structure. Looks sort of like a bent hand, a palm of the hand with these are beta sheets and this thumb-like loop here. Now perhaps because of that and the rigid structure of that leucine-rich repeat structure, these Lamprey antibodies are exquisitely specific, and this one, for example, is tested for reactivity against many different polysaccharides that are put in small droplets on a glass slide. There are 611 of them, and these spikes show the ones that it binds to. And it binds to things that have fucose that has an alpha-1,2 linkage to a galactose that has a beta-1,4 linkage to the next sugar in line. That can vary, but these first two are constant for recognizing by this monoclonal Lamprey antibody. It's the most specific antibody that's ever been seen to this sugar. We've made others that are even more specific, and determined with through the efforts of Ian Wilson's group, which are the important binding sites, and then by the structure-directed mutagenesis, we can make ones that are even more specific than this. So they're exquisitely specific. They're also specific against protein antigen. This is hen-egg lysozyme, and this is another VLRB Lamprey antibody that was made by Zev Panzer, and Roy Mariuza determined the structure of the binding. And you can see this loop that I've been talking about comes down into this cleft, which is the enzymatically active site for this hen-egg lysozyme. These Lamprey antibodies have several features that distinguish them that I've listed here. They're all one chain, one gene, so they're easily engineered. And because we last shared a common ancestor with these Lampreys more than 500 million years ago, they may be able to see things that our conventional antibodies don't see because we have to have tolerance to keep us from acting against self-antigens. And indeed, we've made antibodies that are specific for tumor cells that haven't been seen before. Now, back to the question of which came first, T or B cells. For a time, I thought that shows that B cells came first. But that isn't the case as we look more closely, at least so far, in that there are two other of these incomplete variable lymphocyte receptor genes, VLRA and VLRC. They too are incomplete in their germline status. They too are flanked by multiple donor leucine-rich repeat coding sequences that are used as potential donors to generate highly diverse lymphocyte receptors on lymphocytes. These VLRA and Cs, and ones that the newer ones that I haven't talked about yet, are expressed by cells that have many of the characteristics, and I won't go into all of the data for that look like our prototypic T cells. This VLRA, like our mainline alpha beta, or T cells that express alpha beta gene encoded receptors. These VLRCs are more like ones that use gamma delta in our immune system. We have evidence that they have receptors on them and make cytokines that are counterparts of the ones made by the B-like cells that allow them to interact or cooperate just as our T and B cells do. But our work at this stage is at a very rudimentary stage so far. Our collaborators identified two genes called cytidine deaminase 1 and 2 that are like genes that Jean-Claude Weil has worked with a lot that are responsible for diversifying our antibodies after we put together the V, D, and J sequences in those genes and express them on B cells. They can be diversified through the action of an activation-induced deaminase identified by Hanjo in Japan, and also Alain Fischer's group have shown that in humans it's necessary for generating somatic mutations to have antibodies that have higher affinities or bind better to a particular antigen in a specific response. They are responsible, this gene, AID, is responsible in chickens, as Jean-Claude showed, for a gene conversion mechanism to generate diversity. And they're important, they're essential for switching antibody classes, IgM to IgG to IgA. And Lampreys have something like them, but they have more than one. One of them, called CDA1, is expressed only when in a thymus equivalent at the tips of the gill folds in work that Thomas Boone did with us from Tübingen, and not anywhere else in the body at a time that the assembly of these VLRCs and A's is going on. The CDA2 is expressed only in VLRB lineage cells, and we're still working out exactly when and what all it can actually do. So we didn't find out which came first, T or B cells. It seems that the genetic program for two prototypic T lineage cells and a B lineage cell is a fundamental organizing principle of our adaptive immune system, and it must have already been present in a common ancestor of both the lamprey and hagfish, the jawless vertebrates and jawed vertebrates. But there was evolved apparently as a convergent evolutionary phenomenon of two ways of making these highly diverse antibodies or receptors, antigen receptors, for these T-like and B-like cells in jawless and jawed vertebrates. So this AID, this last gene that I talked about, seemed to have been an earlier component. And there are many other questions, of course, that we would like to answer. But that's about where we are at the moment. These are people who've been involved in this research, and I've mentioned many of them going along. This work was supported very early by Howard Hughes Medical Institute, NIH, and also benefited by Georgia Research Alliance. I mentioned some of my colleagues as we went through with whom I work, and I'd like to thank you for listening to this long, fishy story. Thank you.
Thank you very much. We now have questions. Jean-François Bach.
Beautiful talk, Max. One question, maybe it's an impossible one. Do you know where these B and T cells differentiate? Is there any site or organ that you have identified where these cells differentiate?
We think that they develop the T-like cells in these thymus equivalent regions right out at the tips of the gill folds that I mentioned, and the B-like cells assemble their VLRB genes in this hematopoietic tissue called the typhlosyl that runs the length of the intestine in the larvae and then atrophies. Where the actual immune responses occur and where you get effector development, we don't have a good definition of yet. And we don't have a good idea of autoimmune diseases, I might add.
Pascal Cossard, right here.
So Max, I have heard you talking about the Lamprey, but in other organisms now, where do you find them? Where do you find them in the tree, in the phylogenetic tree? Because are there other people working on this LLR protein involved in the immune systems?
There are people in Japan who are working.
But they are working on the Lamprey.
Yes, and mostly.
But do you find them in other? There is this step that I did not catch.
Oh, I'm sorry, I didn't mention. The same three types of genes, A, B, and C, are present in hagfish as well as in lampreys. We've done a comparison between the Japanese lamprey and the sea lamprey that I mentioned. They are thought to have had a common ancestor about 20 million years ago. The hagfish and lampreys had a common ancestor about 480 million years ago. So all of the jawed vertebrates have this system, the details of which...
I don't know, eggfish, I don't know you translate that. Eggfish. What is it, eggfish? You know?
Hagfish? Hagfish is this animal that ties itself into knots. It's
Mixi?
Yes, I'm sorry.
Maybe. Mixin? I never heard about this one.
Yes. Hans Clevers.
Having worked in the T-cell receptor lab a long time ago, can you say a little bit about how the T-cell receptor equivalents, VLRs, how you think they recognize antigen? Do they see processed antigen, cellular antigens, or?
You ask a question that I can't answer completely, of course. We can easily find the B-like cells that, in small numbers, that will bind any random antigen, protein antigen, let's say, that we pick out and look for binding. After we stimulate them, they're more the frequency of those binding cells increases. So it seems to be clonal expansion. For T-like cells, we've had trouble showing that they can bind native antigens, which might suggest that they have to process antigens in order to recognize them. I thought you were going to ask me how they signal, and we don't know that either. We don't know what the composite structure of these receptors are. The parts that we know about, the antigen binding parts, don't go down through the cell membrane and with intracellular regions that have signaling, recognizable signaling sequences. So we're still trying to find out the overall composition. And
So you think that there are repeats so that the response is stronger? Do you expect the antigen to bind to one repeat, and then the more you have repeats, maybe you have a stronger repeat response?
No, our data suggests that it's more the sequence rather than the length of the receptor. For example, the number of those green blocks in that cartoon, average number of those is 2 in the VLRB, the antibody type. It's about 4 in VLRC. And that gets shaped in the thymus, indicating that they are selected in their development early on, just as our thymocytes are in clones. So I think it's the sequence that determines the binding specificity that from all of our structural studies of antigen binding.
Alain Fischer, down there.
Max, based on what you know about the structure of these VLR receptors, can you deduce some information about the structure of antigens they recognize? Is there a common law on which kind of chemical structure, of what kind of conformation these VLR molecules may recognize?
I don't think we can extend it that far, Alain, but what we can do from the structure is to see, we can design changes in the structure that will change specificity. And we've shown that best for these polysaccharide types of antigens. And we think they'll be particularly valuable in that, as you probably know, the different kinds of carbohydrates and these long strings of sugars that form these complex polysaccharides. We have, it's estimated, over 7,000 different ones of those, different types. And they're not encoded in the DNA directly, so you have to determine them by physical, chemical, or functional means. And we have now identifying antibodies against maybe 5% of those. And so these will be particularly valuable, these Lamprey kind of antibodies, in exploring the and doing experiments with these different polysaccharides, we think, that are used by plants and so forth, as well as we, of course, and often serve as tumor antigens. So we're hoping that they will have useful value there as well.
The last question by Jean-Claude Veil.
Yeah, I want to ask you, these cysteine repeats are very much present in TLR receptor, in innate immunity. Would you say that you also have innate immunity in Lamprey? Would you have innate immunity with something else than the...
Well, innate immunity is I thought always normally used for something you're born with, that you don't have to change the genes or adapt anything. Whereas these are generated diversely after or during development. And and these adaptive immune responses are usually more specific, whereas innates see patterns but not that are on many different bacteria or molecules and so forth. So in that sense, I think these receptors are more like bona fide T and B lineage.
I agree, but you think Lamprey would have also innate type of response? Innate type of receptor or innate recognition?
Oh, do we see those kinds as well? They have toe-like receptors. They have about 11 of them. Not nearly as many as sea urchins, who have hundreds of them. They also have innate lymphocytes. They have a cell that I didn't talk about that looks like a natural killer cell in a mouse or a human. Very, very related. And also one lineage of innate lymphoid cells named ILC2, we can show elements of the genetic program for that type of cell in a specific lymphocyte-like cell. So I think innate lymphoid cells, or at least NK cells, evolved with or possibly even before these specific T and B-like cells.
Merci. Thank you.
If I may, leucine-rich repeats are domains that you find in many, many organisms, even in bacteria. So I think it's just like a domain which can bind things.
Yeah.
Huh?
Yeah.